Yeah, okay. First of all, thank you very much for the invitation and for putting together this very nice program. Um, so, yeah, I'm going to talk about some current work on single-cell RNA data, and most important, most of all, it would be an overview of, like, first of all, the research questions, so what we want to solve, and then our approach. But it will stay very general, and I won't get too much into technical details. Okay, so single-cell sequencing is a very recent method to sequence the RNA or even DNA of cells, and it's called like this because basically, given a population of cells, it enables to extract the information at the level of the individual cells. So if you have a population of cells, you can extract the RNA sequence for each of the cells. And in practice, what we have is not really RNA sequence, but it's translated into what we call an expression vector, and it gives you the expression of some genes of interest for the given cell. So it translates the RNA into gene expression. And typically, number of genes can be several hundred of genes or several thousands of genes. So this is just a representation in 2D of a data set that was recently published on this single cell sequencing. Yeah, and okay, the main question that we have is what they call trajectory inference. So given this population of cells, we want to recover what is the underlying evolution process or dynamic process that parameterize this gene expression. And why is this even a question? Because when we look at this population of cells, it's basically a snapshot. It's just a photography of the tissue at a given time. And at this given time in this tissue, there can be cells in very different stages of the process, but we don't really know which cell is in which stage. And that's basically the question. So given this snapshot, can I recover which cells is more advanced than another cell according to this evolution process that I don't really know? It's just a question of arranging or ordering cells. And it can also be seen as at a more causal level as clustering the cells into, let's say, macro stages. So typically, what we want to look at, maybe it's a bit more concrete, is a tissue that is suspected to be a cancerous tissue. And then obviously in this tissue, there can be cells in different stages of cancer. And this cluster can be stage 1, stage 2, according to the usual nomenclature in hospitals. So the question is just finding this order and not labeling this order, just finding it. Now, what we have, in fact, at least what I have, is slightly more recent. So we have this gene expression, but on top of that, we have another type of data, which is called RNA velocity. RNA velocity is if gene expression is order 0 of our evolution process, RNA velocity is order 1. And it quantifies more or less the infinitesimal change of gene expression for each of the cells. How it's computed, it's a very large question, and I can't really answer that now, otherwise it would take 20 minutes. But it's also its own research question, how to compute RNA velocity out of the RNA sequence of a cell. Me, I just accept that I have this expression vector and I have this RNA velocity. So typically, if I go back to the 2D representation, I have something that looks like this. But now you have to imagine it's like in dimension 4,000, I don't know, something like this. For this data set, it's like 2,000 something. Okay, so now what has been done so far, a lot of things. Most approaches, they are coming from the informatics or bioinformatics community, and they rely on the manifold, so-called manifold assumption. So obviously our data are high dimensional, and that's a problem. But what happens is that not all the cells are concerned with all the genes. So we have very sparse information. And then natural assumption that we want to say, we have the manifold assumption, which is we have a lower dimensional smooth structures that can approximate our data. small structure that we don't know. And then classically, what do we want to do? You want to rely on a neighbor graph to compute some sort of approximation of an intrinsic distance, and then based on this graph, you can do whatever is left to do. So, I mean, very basic approach is like, I compute a neighbor graph based on the distance in R 2000. And out of this graph, I can extract my ordering of the cell. A bit more advanced is, okay, this distance is obviously not very stable, but I can introduce something that looks more like a Markov chain, which is what is my probability to go from this type of RNA expression to another type of RNA expression. And then again, I recover a graph, and out of this graph, I can extract an order. I think this is like a way to summarize most approaches, and then there are more recent approaches in deep learning, obviously, and optimal transport as well. But concerning RNA velocity, I think there's still a lot that has not been really much leverage on this velocity, and mainly the fact that expression velocity is mostly a coupling. So it's not just two independent information, but they really have to do with each other. And this is what we wanted to do. And it took me a while to understand that we have to give up on this manifold assumption because it's not constraining enough. And also it doesn't necessarily make sense, like most of the time. But this time it doesn't make more sense, and it's not even useful. So first step of our approach was to give up on this and to say, okay, we have to make another assumption. And another assumption that we can make is quite motivated, first of all, by biology. So the most common evolution process for cells is called differentiation. And differentiation is basically a tree. It's like, I'm in this type of cell, and now I have the choice to evolve into 3 types of cells. And then maybe this 3 types of cells will evolve in other types of cells, etc. And the like phenomenon of coming back to a previous type of cell is not that common. So it's not the main phenomenon. So we can assume that we have more than a manifold, we have a tree, and based on this, we actually have a much higher constraint that also took me time to understand, but having a tree is nice because it's very constrained, so we can leverage on this. So now I can reformulate the problem of trajectory inference, so ordering the cells into what we call tree inference, because it's how it's called in the literature. I have a set of nodes, which are here my cells, and somehow I want to find a tree that is compatible with this node, so with this embedding of the nodes in R 2000. Yes? But it codes the gene expression of the cell number I. So it's like percentages, sort of. Yes? Yeah, it doesn't say much, actually, but I'm going to explain. Yeah, basically, that's a bit what you want. You want to say, I have an underlying tree. My cells are sampled somehow as node of this tree. And then, obviously, the velocity should give me the direction of the next node in this embedding. But it's very vague on purpose, because then I won't care so much. So. But let's say what I want to find is not a manifold, it's a tree. Okay, so typically this. And this is something I did myself. So it looks very good because I simulated 200 examples and I took the only good one. But that's more or less what I want to do with much more cells. It's not, so yeah, I will comment on that after. So I will decompose this problem in 2 problems. The first problem is I want to learn a metric that is more or less a tree metric. So I have points, I have vectors, I want to learn a distance. When I say metric, sorry, when I say metric, it's a distance measure. It's not a Riemannian metric, just distance. And then when I have this distance, I want to find a tree. And why I'm decomposing it this way is because the second problem is very well known. I give you a distance and I want to find a tree so that the tree distance match my original distance as best as possible. And this is well known, And there are many methods already available to me. Yes.
A distance measured on the tree, so it's like finding a
distance measure. Yeah, distance measure. Even if it's not like proper distance, it's also similarity measure.
And yeah, as I was saying, tree learning, so-called, is very standard, and I'm giving this paper that was published some years ago in Europe with a very branding name, I remember, and but it does exactly what I want. I give it a distance matrix, and it gives me a tree. The only problem is like it's not. You can have many solutions of trees, and these I can maybe make it a bit better because I have information about the direction of my process, which you usually don't have. So I have this vectors, velocity vectors that can also help me saying, okay, actually I have some order already intrinsically. But I will not talk about the second problem now. I will talk about the first problem, metric learning. What can we do with this discrete vector field? We can make it a continuous vector field because it would be much better. And for this, I will interpolate with this kernel regression. I will just interpolate my vector field. And this is already, of course, it works very well in 2D. In high dimension, it doesn't work too well. So there will be a lot of intermediate steps like smoothing the vector field and things like that. But I mean, honestly, it doesn't work too bad. So I interpolate my vector field, for example, with a kernel that is just a Gaussian kernel, and then I choose the sigma, however I can, to have something smooth enough, but not completely erasing my structure, obviously. And yeah, very important, I integrate backward this vector field then. So my cell is the last stage that I have, and I will integrate backward to recover what I call the cell trajectory, which is what is the history of the cell, where does it come from? That's the first step, and then once I have this, to compute my distance, I will compute the distance between the trajectories. So I will compare the curves, basically. And then curve distance, there's huge bibliothèque of them. And I had to try some of them. I mean, not try, like, implement them, but to think about, okay, does it do what I want or not? A lot of them don't do what I want, obviously, because I don't have shapes. I really have, like, trajectory, and the way the change is really the way a tree will grow. So when you grow branches of the tree, it's a very particular phenomenon. So far, the best one that I found in terms of interpretation is the current distance. I know it's very old. I know there's variable distance right now in the market, but I think this one is very nice for what I want to do because I have a very clear orientation and it's very simple, so I can interpret it directly mostly. The only thing that I have to think about is the kernel on the position. So the Gaussian kernel in this case, and how sigma does change my distance basically. But okay, let me explain what I mean by it does what I want it to do. What we want to measure is how two trajectories they branch in between each other. So you can really think of it as a tree, like physical tree, and how a tree would grow two different branches. And at some point they are like the same thing, and when it grows, it will start to divide and to diverge from each other. And this is what I want to measure. And there's two things that I can say. So first of all is like, I want to have a distance such that what I imagine being this branching process, so this branching deformation, I want it to be as much as possible a minimizing path for this distance. And I mean, it's not so hard to check. Like, I just have to define what is this deformation, mostly. And it's related to the sigma in the case of the current distance. And then I have to compute the distance along this path, and then I have to see, okay, it's close to my optimal current distance or not. Turns out it's not so far, so not too bad. And a bit more advanced property, but really related to this, is that I would like the distance to be a hyperbolic distance. So to define a hyperbolic metric space. Of course, because then I want to have a tree. A tree distance is like the perfect hyperbolic distance. So I don't want necessarily like hyperbolic distance with parameter 0. So I don't want a tree distance because maybe it's a bit too much constraint, but something with like delta hyperbolic distance with the delta very small. So hyperbolic distance is just saying, I mean, there's many definitions, but definition that I'm looking for is like if I have a geodesic triangle, so 3 points, and I look at the geodesic path in between the 2 pair of points, I want this geodesic path to be very close to each other. That's what I want. And this geodesic path, they are related to this. So more or less, if I can prove that I have the first thing, then I'm not far from having the second thing. So that's what the current distance do. I'm not yet satisfied. I would like to have something a bit more intuitive. I would like to build the distance itself out of this path, but it's not very, I mean, it's not a diffeomorphic path, so I have a problem here. I can't go to like LDDMM-like approach already, so I have to think about that. But ideally, I would want to build my distance out of this phenomenon. For now, I have the current distance. It does a good job. So it's a bit open question right now. It's like, can I build a better distance that makes more sense from the point of view of the biological intuition, let's say. Okay, I don't know. Fine. Okay, then I can also do a stochastic version of it. And for this, maybe I mean, I could see some faces here that could help me, maybe. But it's really similar solution, similar intuition. So I will replace my OD that I have first with a stochastic differential equation. And rather than integrating curves, I will integrate stochastic processes. And then I think the notion of branching in stochastic processes is quite common. So it's not something completely absurd to think about. But mainly, I think the kind of quantity I want to retrieve is like if I simulate my stochastic process from cell 1 and cell 2, I want to estimate the time when they will meet with each other, stochastic process. And this is, then once I have it, I can retrieve tree distance, more or less tree distance. So this is something that I want to have, kind of hitting time, things like this. And why this is interesting, because obviously it will be more robust to the noise on the velocity vector. And I have a lot of noise on the velocity vector. So that would be a stochastic version of the first approach. But for now, I'm more working on the deterministic approach. And once it doesn't work, I will go to this. Yeah, and then, some few comments on like later questions. So this is more questions that are close to the application and the biology. So as I said, it can also be seen as a cluster problem. So rather than ordering all the cells, I can order like some area of my domain, or I can order some clusters of cells. And clustering according to a tree distance is a bit tricky, because if you apply the usual clustering, then you completely ignore the structure of the tree. So it would be more of from a tree, which is a very fine tree, to go to a coarser tree. And then to, yeah, exactly. So I have this tree of like 2,000 nodes, and now I want a tree of maybe 10 nodes. And my 10 nodes will be answer stages or something like that, like macro states of my process. And this we really want to do that, because also it's what's done in practice. Second thing, we have to work on the visualization, because we want to visualize this thing in the biology community. So there's a lot of tree embedding available right now, but in my experience, they don't work so well if it's very sensitive. So if my distance is not a perfect tree distance, then I don't get, like the hyperbolic embedding, for example, doesn't work so well. So maybe I will have to work on this as well. But this is future questions. Yes,